The study, so the premise for starting this study was data from two other pivotal trials, Sunbeam and Radiance, that observed effectiveness of Ozanimod on cognitive outcomes, particularly on SDMT, and improvement in a large number of patients. And the second reason was that there was actually disproportional effect of Ozanimod on gray matter atrophy, in particular on cortical and thalamic atrophy in both of those studies. And based on that, it was thought that it would be extremely important to see whether changing relationship in cortical atrophy and thalamic atrophy would actually influence cognitive outcomes. So based on that, only a single arm study of Ozanimod has been planned with the enrollment of 250 people with a very early relapsing remitting MS. So the disease duration should have been less than 5 years, and the EDSS should have been less than 3.5 points on EDSS, and people could not relapse or have a high radiological contrast lesion activity at baseline. So it was very difficult to recruit such a young population and a very early population in the study, and it took about 2 years to recruit for this study. At the end we finished about 190 patients because that was sufficient power-wise to show the primary effectiveness on primary endpoint. Now clearly the first primary endpoint, the main primary endpoint, is to show whether Ozanimod can improve in more than 44% of patients cognitive decline on SDMT, and there are numerous secondary and exploratory endpoints, among which are brain volumetry endpoints, including thalamic, cortical and whole brain atrophy, as well as subthalamic nuclei, which may play an important role in cognitive processing. There is also thalamic disconnectivity, which is very interesting new marker showing how network efficiency in the white matter is disrupted or disconnected in patients with multiple sclerosis. Based on this criteria, a patient that is showing baseline relationship between cognitive and brain volume outcomes have been really determined based on specific cutoffs for 3 cognitive tests that we used in this study. So for the SDMT, the cutoff was 44. So everybody lower than 44 was cognitively impaired. For the BVMTR, which is a memory test, the cutoff was 17, and for the CVLT, which is the verbal recall test, the cutoff was 39, and about 20% of the patients the average age was 40 years, and the mean and median disease duration have been about 1 or less than 1 in the study. So very early population with about 40 years of age, about 80% being female, having about already 20% of cognitive impairment. And so what we looked if we divide their whole brain or cortical or thalamic volume in high, low and medium terciles, is there going to be a correlation between cognitive impairment on those tests and the amount of brain volume? And while there was numerically a very good correlation, however, did not reach significance because probably the disease duration was very narrow in this study. But it was all in the right direction, and we hope that over the next 3 studies, which will be the 3 years of the study, which is the follow-up, we are going to see meaningful results.